today I'm just gonna give you an idea about why not just doing brain mapping is not the best thing to do, but how we can define our neural target, how we can get to a level to clarify that what would be the best neural target. Because there are many areas in the brain, there are many networks in the brain. How you can select the best network that works best for you. And we have been trying to also optimize other ways of network engagement and how we might be able to target specific networks involved in craving. And we have been always interested to frontal-parietal networks. For being a successful scientist in the field of human brain mapping, you need to have a career map. You need to know what is going to happen in the next coming years, in the next 10 years, in the next 20 years. And this is where we have started to work in this area. So these figures that I'm showing, these photos I'm showing you, they go back to 2003. So we have started doing this somewhere like 18 years ago, trying to find specific cues that could activate brain areas in people who are using substances. So we tried not to just be stopped at the level of measurement. So we want to go to the next level to be able to develop intervention.
Hello everyone, it is 4:30 a.m. here in Minnesota. I'm really happy that I'm joining you from Minneapolis, and today I'm gonna give you an idea about how brain mapping could be used for developing intervention. So I will try to give you a talk in something like 45 minutes, and then we will have time to discuss about what we have been talking about. Okay, so if you are excited about things like brain mapping, I'm not sure if you see pictures like this and you see brain mapping activations, whether they are from FMRI, EEG, different methods that you use. That is something that I've been always interested to see how we might be able to see what is happening inside the brain. So that is what makes me really, really excited. And this is what we have been doing during last something like 20 years of doing brain mapping studies. And this is a picture from one of the recent submissions that we have done with Ms. Jafakesh from Shiraz University. And so that is what we do. So we are excited about this as well. But I'm just going to change the process and just tell you, please do not be excited about this. Because I think that just being excited about doing brain mapping and being excited about brain mapping is not going to be probably the best way of using brain mapping. And I've seen many people who have been kind of invested their entire life to just to see what is happening inside the brain. But I think that based on my kind of experiences during the last couple of years, I realized that you need to have one more thing to be able to get to a better level of quality, to be able to see what is really happening inside the brain. And today I'm just going to give you an idea about why not just doing brain mapping is not the best thing to do. So that's what I'm trying to give you an idea about that, and trying to tell you that just seeing what is happening inside the brain with brain mapping is not probably the best thing to do. So that is what I'm going to discuss about today. So we'll see how we can move forward for that. And I'm going to give you a talk in 10 major rules. So I'm going to discuss about 10 major recommendations. I think that would definitely be helpful for those, especially who are beginners in the field of human brain mapping. And I think that is going to be something that would be helpful especially for those who are going to be in their first years of being excited about those really beautiful brain maps and blobs of activations inside their brain or inside others' brain. But I hope that is going to be helpful for you to develop a better career in human brain mapping. The first rule is, instead of just doing brain mapping, by brain mapping I mean just developing those beautiful maps of what is happening inside the brain, I highly recommend people to have an interventional plan in what they are doing. So thinking about how they might be able to change something inside the brain, whether the change is going to come from behavioral interventions, electrical interventions, or chemical interventions. Whatever thing that could help you to change the activation inside the brain or the connectivities inside the brain, that is going to be your interventional plan. And the reality is, When you have an interventional plan, then you would be able to understand what is happening inside the brain, because most of the studies that we do with human brain mapping are mainly correlational studies. But if you really want to know what is happening, you need to have a causal understanding about the brain. And to have a causal understanding about the brain, you need to be able to change it. So if you are able to change something in the brain and see, okay, if I change this, can I change the brain mapping that I had before? That would give you a higher level of understanding. That would give you a causal level of understanding. And that is going to be something exciting about brain mapping. And without being able to do any sort of intervention, you will never have that great opportunity. So that is the first recommendation. Try to see how you might be able to have an interventional plan in whatever you do in human brain mapping. That is the first item. The second item, I highly recommend people to pick a specific battlefield and try to get focused to a specific human cognition or behavior that they might be able to modulate. So trying to see what would be the best field that they can invest their career for. And let me just give you an idea about what are the potentials in terms of how the brain is functioning. So if we go for major causes of death among human beings, this graph is from a recent publication in JAMA, and I'm gonna kind of ask my colleagues, I'm not sure if they are here, but I was kind of asking them to give you links of the publication I'm sharing with you, and I hope that would be happening as well, so you can kind of see the links of the publication I'm discussing about inside the meeting room with my kind of colleagues who are sharing those links with you. Meanwhile, I will share the slides, and I put all the links of the kind of items I'm presenting in the slides. So what you see inside this slide, let me just see if I can bring up my pen as well. Okay, see, I have the pen. Okay, so what you see here, you see the major risk factors, okay, of this. And you can see here the number of deaths that they are causing inside the United States, okay? So when we are talking about something like 500,000 deaths per year, okay, just inside the United States. And when we talk about other countries, it's close to that. So if you go to other countries around the world, you can see that the risk factors are almost the same. So when we talk about risk factors, we are talking about dietary risk factors, how people eat and what people eat. Tobacco use, having high blood pressure, being obese, having high blood glucose, having high level of cholesterol, alcohol and drug use. So you see many of the risk factors that contribute to millions of human deaths over the time, they are modifiable. So they are what they call modifiable risk factors. So we can modify them. And if human beings start to be in control about what is really happening in their brain, or if we provide them with specific sort of interventions about what is happening inside their brain, they are able to control all these risk factors, and then we can stop many deaths around the world. So then you can start to think, but okay, is there any chance that I can be involved in this process of helping people to change what is happening in their brain in a way that we can reduce the number of deaths? So that could be one of the good justifications to decide about what could be the area of brain mapping or area of, let's say, cognitive neuroscience that could be interesting for you. And I might be biased because I've always been interested to areas like obesity, addiction, how we might be able to help people to change their behavior. But that is the battlefield that I decided to go for, because I realized that this is something that people are strongly need around the world, and that would be something that would be definitely helpful for the human being. So that is my justification to go for the areas like eating behaviors, addictive behaviors, and things that we can change in a human being. But that might be, I mean, my own kind of justification. You can go for other justifications that would be helpful for you guys. But decide about your battlefield and think about what could be the best area to do these modifications. And then I have the color code for these kind of beautiful figures that you have about different type of disease that they are contributing, the disease factors, how these risk factors contribute to different diseases. And you know many diseases that we know around the world are being affected by all these risk factors, okay? The third item, define the neural target that you are interested to. So think about, okay, I'm interested to that specific network, that specific brain activation. And thinking about, okay, when I'm talking about an intervention or thinking about having an intervention for yourself and trying to use that intervention to change an outcome, what we usually have a target. And the target is a brain network A, brain network B, whatever what we call it, or brain kind of area A, B, C, or whatever we are talking in terms of the naming. So it could be, let's say, frontoamygdalar network. It could be frontoparietal executive control network. So it could be whatever you are interested to. But when you try to define a specific neural target for yourself, then you can get focused on a specific target and learn about that target and try to understand what is really happening on that target. And trying to see what are the interventions that could help you to modify that target to get to a specific outcome. And the outcome could be lower calorie intake, like what we have been discussing in terms of changing all those risk factors with helping people to lower their calorie intake with any different sort of interventions. So you can put whatever brain network that you are interested, whatever battlefield that you are interested to here, and different types of interventions that could help you to modify that specific neural targets. So this is my main recommendation here today. Just start to think about how you might be able to do that in terms of having an intervention, thinking about a specific battlefield, the outcome, a specific area of interest, whether it's a specific disease or a specific behavior that you are interested to, and also thinking about how you might be able to have a specific neural target that is going to be contributing to this entire story that we are discussing about. And then you can start to think about, okay, when I do an intervention, is there any hope that I can engage that target? And what do we mean by engaging a target? By engaging a target, we mean that we are able to change brain network connectivity, for example, for this one, from one state to the other. So we are able to change what is happening here. If it is a specific area, let's say medial amygdala. When we talk about medial amygdala, when we are engaging medial amygdala with a specific intervention, then we need to be able to show that we can change the activation in medial amygdala from, let's say, 2 to 3, from one number to another. So we are able to engage that target. And target engagement is a key term here. And if you are able to engage a target, that means that you have a higher level of understanding about that target. So if you are interested in brain mapping, if you are interested to see all those beautiful figures from what is happening inside the brain, you need to show, okay, now I'm also able to change it in a direction that I like. Okay? And then the next question would be, okay, the target is engaged. But when I change the target, when I engage the target, can target engagement result in a specific behavioral or outside outcome that I'm expecting? Let's say people are starting to have lower calorie intake. Okay? Then we say that this target engagement is valid. So you need to learn about two key terms: having a target engaged, and then being able to see that target engagement could change something in external behavior, which we call it target validation. So you need to have target engagement and you need to have target validation. So these are two major things that you need to be able to do. And to have a better understanding about this sort of model or a statistical model that we are showing here, you need to learn about what we call mediation analysis. And if you have never done mediation analysis, that will be something that that will be your homework. You can go and kind of learn about mediation analysis. And there are sometimes partial mediation analysis or total mediation, kind of partial mediation effect or total mediation effect. If you have total mediation effect, you technically do not have any effect here. The effect would be just mediated through this pathway. And that is something that you can learn about that. But that is not our discussion right now today. We are not going to discuss about the statistical analysis. I just wanted to give you an idea about what is really happening here and what are the structures or basics that you need to learn today in the next couple of minutes. Okay. Then the question would be, okay, as we discussed, you need to define your neural target. But how we can define our neural target? How we can get to a level to clarify what would be the best neural target? Because there are many areas in the brain. There are many networks in the brain. How you can select the best network that works best for you? Okay, so that is the question that we need to answer. For being able to define the neural target of interest for yourself, you need to have another level of understanding, which we call neurocognitive mechanistic models. You need to have a neurocognitive mechanistic model. And what do we mean by neurocognitive mechanistic model? When I'm talking about neurocognitive mechanistic model, let me just give you an example. For example, when we are talking about a person who sees let's say a chocolate cake, okay? And then the person starts to eat it, or the person decides not to eat that piece of chocolate cake. So there should be lots of things happening inside the brain. When you are exposed to having a piece of chocolate cake in front of you, and then you are deciding whether you should go and eat it or you decide not to eat it, okay? There should be many things happening inside the brain, and there should be many activities or connectivity inside the brain involved. How we can summarize them into a simpler model? This is what we do in terms of seeing what are the cognitive functions involved. And I'm not going to discuss about all the cognitive functions that are involved, but there is a dynamic process happening inside the brain. And here with this specific model, we try to summarize them. And also we discuss about what are the brain networks involved in all these steps of cognitive processing in the brain. So then we call it neurocognitive mechanistic model, because that is the mechanism that drives the process inside the brain, okay? So you need to have this sort of simplified model about what is happening inside the brain. And based on this mechanistic model, you can start to say, okay, it seems that I have dorsal attentional network, I have salience network, I have default mode network, I have control network. And then starting to realize that, okay, what will be the best network that I can be focused to as my main, let's say, neural target, okay? So you need to pick your neural target or neural targets based on these neurocognitive mechanistic models. And there are many of these neuromechanistic models available outside in cognitive neuroscience. So if you're interested, you need to learn more about cognitive neuroscience, and you can learn about all these different networks that are available there. And these networks even could be really simplified in terms of the different cognitive processes that are involved. This is another depiction of the same model in a more, let's say, humorous way, to give you an idea about the processes that are involved when you eat something. So how environment contribute to that, then attentional processing, memory, saliency processing, what we call interoception, control, and then response, how these processes work together. So this is something like a really simplified, this one is, I can say, a cognitive model, not neural, but it's a cognitive model that describes the cognitive processes that are involved. We published that specific model that we call an easy core model in a paper, not recently, something like 4 or 5 years ago, but you can learn about that specific model there. That would be something that you might be interested to see. And we have published that with a couple of colleagues, with Padide, Fateme, and Dr. Mokri, who's a kind of one of the serious figures in the field, and John Montroso, who is one of my friends in USC. So, and Padide is a PhD student in USC right now. So with a couple of colleagues that we developed this model, and you can learn about that in that specific paper. Okay, and in that specific paper, we discussed about basically circuits. So when we talk about circuits, we talk about networks. And then interventions that could engage these circuits, okay? With the hope to have an outcome of being able to modulate drug craving, to see how we might be able to change people's behavior in terms of not to take drugs. So that is how, we summarized this process in one paper in terms of discussing about interventions, targets, regarding the outcome of interest that we are interested to, okay? Fifth item. In the next step, you need to get to another level and see how I can measure target engagement. And this is the step that FMRI kicks in. And now we need to discuss about, okay, I'm interested to this model, I'm interested to this specific neural target. I realized about the interventions that could engage that specific target. But the question is, how can I measure target engagement? And this is how we use fMRI. We use fMRI to measure target engagement, to measure how I might be able to change a specific target, okay? So this is what we call target engagement measurement with fMRI. And you need to be really good at that, really, really good. It's not just about, okay, I know that there is an fMRI task, I can show people those pictures, and then I would be able to see what is happening inside the brain. You need to be way better than this in terms of really understanding how you are going to measure that specific neural activations or a specific network activation inside the brain. And let me just give you a few examples about that. For example, when we talk about things like affective or appetitive areas in neuroscience, as we have been discussing about things like eating or using drugs or being addicted to social media or being addicted to gaming. Whatever we are discussing about regarding what we call appetitive behaviors. And also when we talk about affective behaviors or affective neuroscience or affective neurotargets, things like emotions, fear, anxiety, depression, and all the other things that would be associated with that, like anger, elation, happiness, whatever that would be related to affective processing. So when we talk about this area of neuroscience, most of the time we have a cue. So we use a cue to activate those affective or appetitive neural targets. And that's the reason that we use these cues for fMRI to be able to measure those activations inside the brain. And this is where we have started to work in this area. So these figures that I'm showing, these photos I'm showing you, they go back to 2003. So we have started doing this something like 18 years ago, trying to find specific cues that could activate brain areas in people who are using substances, and those brain activation would be involved in kind of their addictive behaviors, okay? So to be able to measure that accurately, we have started to develop a kind of a series of drug-related cues, and we have been asking people to see those drug cues and rate them, and try to see how they could engage, their drug craving. And to be able to do fMRI with that, we had to also have neutral cues as well, because that is what we do in fMRI. We do subtraction. We just expose people to drug cues, we expose them to neutral cues, and then subtract the brain activations to find areas that are specifically activated because of those drug cues. And then we actually need to, when we want to design an fMRI task, we needed, for example, for each blocks of drug cue or neutral cue exposure, we needed something like 6 pictures. So we need to have a pictorial database, okay, to be able to measure that inside fMRI. And that was the reason that we decided to have a large database of drug-related cues. And this is what we have developed. I'm not going to go to the details about that, but I mean, we have been really successful in terms of defining a specific, for example, blocks of opioid cues. As you see, we have 6 cues here in each block, as I showed you in the fMRI task. So 4 blocks of drug cues and then 4 blocks of control cues. And all the things are matched in both sides in terms of brightness, all the psychophysics features. But the only thing that is different is the content in terms of being drug-related or not drug-related. So that is how we control these figures and these pictures. And we published that database recently, so the database is publicly available. But we tried very hard to make sure that this database is something that people can use. And now people around the world in different kind of countries, in different labs, they are using this database for developing new fMRI tasks. But you need, if you want to be an expert in neural target engagement measurement with fMRI, you need to have a really good understanding about the task that you have and try hard over the time to excel in developing that fMRI task and being able to use that fMRI task. And beyond developing that fMRI task, that you need to invest. As you can see, we have been investing on that during the last 18 years to just make sure that we have the best possible tool to engage those neural activations or to activate those neural activations. And then you need to also be able to do studies in terms of using that fMRI task and see how you can engage different brain areas. And there are different ways of showing that. For example, this is the activation in different areas in Brain Autom Atlas that we usually use. Just give you that really beautiful brain map. But before being able to do that, you need to kind of be sure about the fMRI task that you are using, okay? And beyond that, you need to also, it's not just about the average activation that is happening inside the brain. You also need to have a better understanding about temporal dynamics. It's important for you to have an understanding about, okay, how these activations would be changing over time. So then we start to have another factor, which is time. And showing these blocks over the time, here we are repeating them for four times. They are not identical cues. They are just cues that are equal in terms of their potentials for inducing drug cue reactivity, but they are not identical. So we just do not repeat them, but we just have 4 times of these cues, and then we can see how different brain areas would respond to these cues over the time. So then you have a better understanding how these areas are engaged in that specific task. Okay, for example, here you can see that areas like kind of inferior frontal gyrus, right and left. I mean, right inferior frontal gyrus, left inferior frontal gyrus. You have a sort of a stable, this is drug, this is neutral. So you have a stable activation drug versus neutral in these areas. But there are areas like, let's say, ventromedial prefrontal cortex or right amygdala that we have a rapid habituation. So in the drug-related activations would just get habituated really rapidly. So you technically have just activation in the first few exposures, and then the activation would be gone because they dynamically habituate to that exposure, okay? So then you can start to not only just seeing an average, but also seeing what is happening over the time in that temporal dynamics, okay? So then you can see, okay, now I really know what is happening in the brain. And then you start to realize, okay, now I need to go to the next step. I know what is happening inside the brain when I expose people to drug cues. Now I need to realize, okay, how I might be able to change that. That is the time that you need to start to think about how you can have a specific intervention, okay? This specific result has been published in Neuroimage, so you can find them in Neuroimage. They have been recently published, so you can find the result in Neuroimage. Now you need to go to the next step. Now you need to think about how you can develop your own intervention, how you might be able to change those neural activations that we have been talking about, okay? I have seen many people who stop in this point in terms of they have been just measuring these activations and these target engagement over the time without having a specific intervention. But I highly recommend you to think about that in terms of, okay, how I can have an intervention to be able to change those neural activations or neural connectivities. To start to think about the interventions that you can make, there are different models, there are different groups of people that are developing these sort of models and ways of developing intervention. I really like what people are doing in a society that we call Society of Behavior Change, what they do in terms of the kind of what they call science of behavior change. So you can find their website is called scienceofbehaviorchange.org, and the way that they are discussing about is that would be interesting. So I'm going to give you an idea about how to start to think about developing interventions. Here we are talking about behavioral interventions, things that could help people to change their behavior, not based on brain stimulation or sort of chemicals that we give people. It's just about cognitive or behavior interventions that we provide people, okay? So when we talk about behavioral or psychological interventions, there is always a black box. So we have an intervention, there is a change in behavior. What is really happening inside here? And this is where you can come inside and say, okay, I'm a cognitive neuroscience expert. I can measure neural target engagement. How can I open this black box and tell you what is really happening when you do a specific intervention that would contribute to changes in the behavior, okay? So we are going to open this black box together. And to be able to open this black box, people decided to provide specific methodologies to make it possible for people to share what they are doing. So that is going to be something that would be interesting to learn more about. And this is exactly what we have been discussing about in terms of most of the time, those who are doing psychological interventions, they know, okay, there is an intervention, there is an outcome, and that is what I know. But they really don't know what is really happening in this black box. And this black box is the area that you can kick in as an FMRI expert to let people what is really happening when you do a specific intervention. So then you can start to collaborate with those who are right now in what they call interventionists, those who are doing interventions, and help them to see what is happening inside this black box in terms of neural target engagement, okay? Now you need to start to learn about a specific approach that we call experimental medicine approach, okay, EMA, experimental medicine approach. So we are learning about this specific approach. When we talk about experimental medicine approach, we need to identify mechanisms. We need to measure those mechanisms. And when we talk about mechanisms, this is exactly the neural targets that we have been talking about. So this is neural target. When we measure these neural target, this is where we have FMRI or EEG or other human brain mapping methods. And then we measure if we can engage or influence that mechanism. And that would eventually give us a mechanism of change. So this is what people are really interested to see. how they might be able to have a mechanistic understanding about what is happening inside the brain. And why it is important to have a mechanistic understanding, I will tell you.
So when we do experimental medicine approach, as I told you, we need to, that is the sort of a classic textbook definition of experimental medicine approach. When we do experimental medicine approach, we need to specify mechanistic target of intervention first and define specific methods to engage those targets. Okay? If we do a specific intervention and we have these things in terms of specified mechanistic target and a way to engage those targets, that is what we have in terms of having an intervention in the level of experimental medicine approach.
Okay, just to give you an example, that is what we have been doing recently in terms of, not very recently, but trying to see, okay, how we might be able to develop different sort of interventions. Trying to see if we can have a specific intervention, how we might be able to modulate those activations that we have been measuring with fMRI. So we tried not to just be stopped at the level of measurement. So we want to go to the next level to be able to develop intervention. And we have recently, there are different examples of what we have done in terms of developing interventions, but one of the recent interventions that we developed is called CREF. We have developed with a group of colleagues. Parnion, you can see Parnion's photo here. We have Parnion, we have Tara involved, and we have Warren Bickel, who is one of the famous figures in the field of the temporal-based interventions. And I'm not going to go to the details of the intervention that we are developing, but probably one key term that might be interesting for you is the cue. So we develop interventions based on cues. Here we are talking about substance use disorder, but could be used in other areas. Trying to change that specific cue reactivity, and we have specific neural targets for that. So we go for specific neural activations on the map that we have been discussing and develop a specific intervention. And there are a specific sort of cognitive processes involved in new intervention that we have recently developed. But I'm not going to go to the details. But it would be interesting if you are interested to this area to just go for the details that we develop in terms of how we can engage those neural targets and cognitive targets that we are interested to. Okay? So now you need to kind of go to the next level. Okay, now I have measurements. I just started to develop interventions, and now I'm going to go for the last few remaining steps. One of the other things that you need to know is that, okay, I have a specific intervention to modulate that specific neural target I'm interested to. But what are other ways of doing that? What are the other interventional methods that could help you to engage that specific target? For example, right now we are talking about a specific intervention. For example, we are developing an intervention which is called CREF, okay? It's a behavioral interventions. But is there any other intervention to engage that target? For example, is there any medication that could contribute to that neural target engagement? Or any neuromodulatory intervention, whether it is TMS, TES, any other neuromodulatory intervention that could be contributing to that neural target engagement. So how can I have other areas of intervention being involved in neural target engagement? Okay, so you need to think about other ways of doing that. Why it is important? Let me just give you a few examples. I will discuss about that in the next steps. For example, we have always been interested to see how we can use transcranial electrical stimulation. Okay, and to be able to have an understanding about how transcranial electrical stimulation could contribute or could engage targets, we need to do that inside fMRI scanner. Okay, so we started to develop what we call TES fMRI studies. And one of the kind of figures that we have here is, Zainab, who was working with me before. Now she works with Maron Bixon in his lab. And this is one of the joint publications that we have published in 2019 about how to integrate direct current stimulation with fMRI. And there are technical details on that paper if you are interested to learn about. But that is what we have been trying to do in terms of engaging, kind of trying to understand what are the other ways of engaging those neural targets that we are interested to developing methodologies for that. And then we have another recent publication discussing about all studies that people have done with combining fMRI and transcranial stimulation and the parameter space about that. What are the methodologies that people are using? This is another collaboration with a group of colleagues, Fateme, Asif, and Payman, who have been contributing to that. And then another recent quality control checklist for what we call concurrent TS fMRI. So we do TS fMRI, we do TS inside the scanner while we are measuring neural target engagement with fMRI. So that's the combination that we have recently been working on, and the kind of a champion of that collaboration is Payman Gobadi, who is now a PhD student, and he's doing a really good job for that. So we have a large group of colleagues. We put together a consensus protocol. It is under review right now in Nature Protocol. So hopefully we can get a good review result and we can publish that checklist soon. But the checklist is available on medRxiv right now. So if you are interested, you can find that on medRxiv and you can learn about that. This is exactly what we have been trying in terms of, seeing what are the other methods that we can use for neural target engagement. And there are some technical details about that. So those who are interested, they can go and see those technical details in the publication I've been discussing about. And we have been trying to also optimize other ways of network engagement and how we might be able to target specific networks involved in craving. And we have been always interested to frontoparietal networks. So we developed specific methods to make sure that we can target these neural networks inside the brain. And we have recently submitted a new work by Ghazal Soleimani from Amir Kabir University. I mean, she's doing a fantastic job for that. And you can find that on biomarkers. It is still under review in human brain mapping. So hopefully we can get that published as well. But trying to see, how we can have other ways of neural target engagement and see if we can have a causal understanding about those neural targets that we are interested to. Now as we get to the next levels of having a mechanistic understanding about what is happening in the brain, we can technically combine those interventions together. So we can combine, because we know what we are doing in the brain. So when we start to have a mechanistic understanding, when we start to know how we engage different areas in the brain with behavioral interventions, with medications, with neuromodulation, then we can start to combine them because we know what we are doing inside the brain. We know what are the targets that are engaged with these interventions. And this is the best thing that you can do in terms of seeing how you might be able to put these different interventions together to develop a more robust interventions in the brain. And to be able to do that, you need to have that mechanistic understanding. You need to know, how all these type of interventions are engaging those neural targets and what are the neural targets that are involved, and then try to see, how can I combine them together? And then you can technically optimize the dose of intervention. Even you can start to see how people are responding differently because you know the mechanism. You know, if I'm doing this specific intervention, I'm changing, for example, medial amygdala activation. And then you can see, there are people who do not have that amygdala activation. So you can predict that those people would not be responders to that specific intervention. So you can start then to realize, what are the differences between people and what are the kind of the next steps. And I know I'm aware of the time limitation. I will be kind of finishing something like less than 5 minutes, so making sure that it would be done. So you can have this, consider these inter-individual differences in the model that you are talking. And then you can have what we call multi-level integration. And that is the ultimate goal for us. That would be the best thing to do, to have multi-level integration. And to be able to have a better level kind of understanding about multi-level integration, it's always good to kind of learn about things that people are doing in this area. And it would be nice if you go for what we call RDoc. The good thing about RDoc is that they are trying to document all these multi-level integrations, how we can kind of connect all these dots together to have what we call multi-level integration. And then you can go and develop what we call develop your own biomarkers. And what do we mean biomarkers? Biomarkers, I mean, then you know, okay, if I have a specific intervention, I can change this specific brain activation from this to that. And then you can consider that changes in brain activation as a mechanistic biomarker of response to this specific treatment. So we call it mechanistic biomarker or treatment response biomarker, okay? You can also have what we call predictive biomarkers in terms of, okay, if I measure activation in this area, the level of activation in this area would predict how my intervention is gonna change people's behavior. So we call them predictive biomarkers. So you can have mechanistic or treatment response biomarkers, or you can have predictive biomarkers. And this is the level that you can get and having FMRI as a biomarker, okay? You need to master in literature. You need to see what people are doing. You need to learn about what people are doing. And if you are interested to that, it would be really nice if you start to see the studies that people try to use FMRI as an outcome measure, as a treatment response biomarker for clinical trials. And people are doing that extensively recently in terms of using FMRI as a measure in clinical trial to develop interventions. And that is something that would be interesting to learn about. And that is one of the recent studies that we have published with Alale, one of the other PhD. She's right now a PhD student in Ruhr in Germany. And we have recently started to kind of put them together in something like a systematic review of FMRI in behavioral interventions. We call it FEB project. A group of nice medical students from Mashhad University are contributing to that. I'm not going to go to the details, but if you are interested, you can be in touch with Dr. Muhaddes Ardebili, who is leading that project. But that is another kind of level of collaborations that we are doing to have a better map about this. And the last item. For being a successful scientist in the field of human brain mapping, you need to have a career map. You need to know what is going to happen in the next coming years, in the next 10 years, in the next 20 years. And I highly recommend you to consider interventions as a part of the map that you would be interested to. That would definitely be helpful for developing a map that would be also not only exciting for you, but also exciting for granting agencies, for people who are working outside, and also being able to help your society and your community in long term. And there are many potentials for that. Okay, let me just conclude. We have discussed about 10 basic rules. We have discussed about having an interventional plan, picking your battlefield. What is the area of what would be your area of being focused and interested to? Define the neural targets, the neural network that you are interested to. Select the neurocognitive mechanistic models that are interesting for you and help you to select your neural targets. Try to be really good in measuring the neural target engagement with the FMRI and try to learn whatever that is related to that. Develop your own interventions. Try to see what are the other ways of engaging that neural targets. Go to a level to be able to show that this specific fMRI measure could be a predictive or a treatment monitoring biomarker. Be master in literature and know what people are doing in this specific area, and figure out a long-term career map for yourself. trying to put all these pieces together. And in the last slide, I knew when I'm talking about kind of all the things I've been doing during the last 20 years, I definitely need to thank all mentors, supervisor, professor that I have been working with during the last 20 years. And I provided a kind of few photos from those I have been learning from. And I'm also still waiting for the next mentor that I can learn from. So being in learning process is something that would definitely be helpful for you. And I highly recommend you to acknowledge every time, whenever you do whatever you do in the field of science, to acknowledge all the supervisors, all the kind of senior figures that help you to get to this level. And I do the same every time, kind of thanking all these really great figures in my scientific life that they helped me to get to the next level. Thank you very much.